So my presentation will be divided into four main parts. In the first part, I will introduce the technique, and thanks to Dr. Yassin that saved my time on a lot of concepts. Next, I will talk about the clinical candidates from artificial intelligence native startups, which we said, artificial intelligence focused startups. And then I will stick on the RLY 1971, which has ability to leapfrog to the clinical trials in just 3 years. And finally, I will disclose one of our projects in RugMed, where we use artificial intelligence to accelerate our drug discovery process. So why we need the artificial intelligence in the drug discovery? Actually, the very expensive price tag of the drug discovery process due to high failure rate during the clinical trial necessitates the introduction of artificial intelligence, which save around 70% of the cost and narrow down the time frame for the drug discovery. So we can say that artificial intelligence provide a cheaper and more effective drug discovery process. So artificial intelligence can help the pharmaceutical sciences in many ways. Actually, it can help in the clinical trial design and in pharmaceutical manufacturing, development, and management. And in the drug discovery, it can help in two ways. First, in the design of the active modalities by target identification and drug target binding and the de novo drug design. And it can also help in the screening of massive library of compounds to give us a hotspot in a short period of time. It can also predict the bioactivity and toxicity and physical chemical characters of our chosen compounds. So we can say that the artificial intelligence reshaped the drug discovery cone. So instead of spending a years in the first era of, or the first area of the drug discovery, trying to try different hits and different leads and try to and have kind of trials and errors, which like kind of time consuming and can take around from 7 to 10 years to broaden the probable or the potential compound to the market. We can broaden the first step or the first stage by using ultra large library of compounds, so we can get our drug-like molecule in just 1 year, and then we can move confidentially to the next step, and finally we can get our drug to the market within 3 to 5 years. So actually the using of artificial intelligence in the drug discovery started as early as 2010, when a giant pharmas like Pfizer, Novartis, Sanofi, and others tried to make an active collaboration and active partnerships with different artificial intelligence companies that focus on the drug discovery, like Exscientia and Benevolent, and others that not focus, like Microsoft and others. So these active partnerships actually was aided to get a potential target and unexplored target for complex and multifactorial diseases. And this actually triggered a wave of what we said artificial intelligence native startups that I will talk about in my next part of this talk. So in order to get a very good or a very applicable drug artificial intelligence method, you need to have a good generative model, as Dr. Yassin said, which is powered by automated reasoning. So the thing is very easy, but it needs a lot of money and a lot of assets to do it. So you feed your computer the data, you have a generative model, and it will give you a conclusion. And this is the key of your drug discovery process. So here in this diagram, I show all the process in a very simple way. So any generative model, which is composed of encoder and decoder, is composed of or really on a massive or ultra large library of data set of different disciplines, include medicinal chemistry, gene therapy, biomedical science, and biochemical science. And all these data will enter into kind of a mixer that provide kind of pricing and exploring. And finally, you can get a very useful information regarding our process. This information involves target identification and binding affinity and the binding site of the compound itself and some other conclusions about the toxicity, solubility, and drug likeness characters of our compounds. So in order to get a solid and very applicable artificial intelligence method, the artificial intelligence research engineers should come in contact with the chemists, which include medicinal chemists and biological chemists and computational chemists, and also come in contact with biologists on the other side, which is the basic science biologists and applying or clinical biologists. These meetings lead to formation of an applicable method, applicable artificial intelligence method, and also to remove any doubts that the compound that would be suggested by the machine would be a drug. Integration of this data into a knowledge graph with some other iterative data or internal data that generated inside the company itself can provide unique insights, which inside the wet labs, inside each of these startups, can be converted into a real science or a real hypothesis that can be used in the drug discovery process. So in this part, I will focus and enumerate the most prominent artificial intelligence native startups that were able to bring their compounds into the market in less than 3 years. And this slide showed you the most important 6 companies. And if you read about their stories, you can say that you can see that the stories are kind of similar. But if you look at the details, you can find kind of tiny things that distinguish one from the other. So each one of these companies were able to drive a hypothesis-driven artificial intelligence discovery process inside their sites, and were able to build their own platforms, which are composed of clusters that filled with a huge amount of data. Finally, they were able to build a high accurate decisions based on their calculation inside the company, and accurately define the complex diseases and find effective drugs for these diseases. So I will move one by one, and then finally I will stick on the Relay Therapeutics and its drug candidate. So moving into Essentia, Essentia has a strategy of target selection and applying artificial intelligence to completely identify the target and move forward to get the drug. So the first step in any Essentia research, like the things that they published already, is to confirm the drug disease target association using a generic data and a massive data that's already published online. And then by using this data, they go forward to record the response of this target to their molecules using a real patient samples that allows them to accurately define the potential patient response in the future. And finally, they go into a round of target validation and to assign the druggability and trackability of their molecules. And with this loop in the left side, they were able to bring 2 compounds to the market. The first one is DSP-1181, which is kind of similar to haloperidol for treatment of obsessive compulsive disorder. And they were also able to bring the compound EXS-21546 as A2AR receptor inhibitor for immuno-oncology. Moving to the in silico, our collaborator in RugMed, in silico empowered by a very solid platform, the Panda Omics platform, which contain around 5 million omic database samples and 1.3 compound biologists and around 3 million grants that already got accepted from the NIH and other foundations. Also, it contains around 3.8 million patents and more than 300,000 clinical trials and 3 million publications. They use this huge or this solid platform to move their discoveries forward. So they started with target exploration, followed by evaluation, and then they use an add-on inside this platform called Chemistry 42 to choose the best fit compound to their target. And finally, they do a kind of simulation to the clinical trial, and this is something that makes in silico unique. They do kind of simulation of clinical trials inside their clusters. Finally, they were able to bring 2 compounds to the clinical trials, compound 8 as toll-like receptor inhibitor for treatment of rheumatoid arthritis, and compound ISM001005, which they didn't disclose its structure, is the first in class for treatment of idiopathic pulmonary fibrosis with target X that they didn't also disclose for an intellectual property purpose, I think. Moving forward to the Recursion company, which focus on the rare cancer condition. It used, or this company used Recursion operating system, which is kind of a closed loop of kind of identification and then validation. And using this closed loop, they were able to identify unknown targets for the first time by using empirical data generation, followed by advanced computational chemistry that they lead. They have one of the best computational chemists in the world. And based on this, their computational calculations, they built a novel insights that nobody reached before. And finally, they started the discovery initiation, or they started the discovery work based on what the novel science they generate. So far, they have 3 molecules in the clinical trials, even though they have a lot of compounds in their pipelines, but those 3 were able to move to the next stage. Compound REC-3559 for gangliosidosis, and compound REC-2282 as HDAC inhibitor for treatment of neurofibromatosis, and compound REC-4881 for treatment of familial adenomatous polyposis, which is kind of a rare cancerous diseases. So in company Ferus is a South Korean company. They adopted the ordinary way of the drug discovery that we use every day in our laboratories, but they empowered it by their ChemVerse platform, which is kind of a limited platform. They started with the best target identification, which is the one that's already familiar and already known, and used it to do a ligand-protein interaction using their add-on DeepRecon. And from there, this data, they can assign or get an idea about the candidate substances that were able to go to the next generation using the ChemGen. And also they have a small add-on that can assign or can predict the ADME properties and physicochemical characters. So they can move from this step to the testing step in a very solid base. So far they have two molecules here also in the clinical trials, compound PHI 101 as for treatment of triple-negative breast cancer, and compound PHI 301 for treatment of ovarian cancer, that they also didn't disclose its chemical structure. Benevolent here have a very solid platform and one of the oldest platforms in this industry, and their strategy is to optimize the earliest and most critical phase of the drug discovery process. So they started with exploring of the mechanisms of multifactorial diseases that we already know that has kind of multi-factors or multi-markers that can control this disease. They have a big data from 85 sources, and based on their identifications or their theories, they can move to validate the novel drug target based on what they get in the first step. In this part, they use kind of cutting-edge technologies like CRISPR-Cas9, RNA sequencing, and use pluripotent stem cells to do their experiments. And from there, they can move to the wet lab experiments in their companies using cell-based model, and from there, they can build their own molecules. They have two molecules in the clinical trials that they didn't reveal their structures, compound BEN-08744 and compound BEN-2293, and they also, in an active collaboration with AstraZeneca since 2010, to identify 5 novel targets for treatment of cancer diseases and other diseases like Alzheimer. So in this part, I will stick on compound RLY1971, which were able to leapfrog to the clinical trials, as I mentioned earlier. This compound from Relay Therapeutics, the Cambridge-based company. This compound is allosteric inhibitor of SHB, the oncogenic protein, which has kind of a very important role in different types of cancer. This compound is currently in phase 1 clinical trial, and we're able to beat the Novartis compound, as I will show you in the next slide. And actually, the best or the most promising data of this compound encouraged Genentech to make kind of an agreement with Relay. They paid kind of upfront $100 million, and they will pay another $600 million when the compound enter to the market. Because Relay is kind of a limited company, they will not be able to do the pharmaceutical industry part and redistribution worldwide. So as you can see here, the compound can bind to the SHP1 and can prevent it from going from the dimer to monomer state, and so it will inhibit its action and keep it or sequester it in the inactive form. As I said previously, Novartis was earlier discovered one-hit molecule that has the ability to, and it was the company that firstly explored the SHB2 function, and using high throughput screening campaign, they were able to discover this hit structure in around 2012, and from there they moved to the lead and to the clinical candidate in a like kind of a classical manner. And in 2016, they were able to identify compound 2NO155 as a drug-like compound to inhibit SHP2. The IC50 of this compound was 11 nanomolar, and remember this number well. So in Relay Therapeutics, they used a different way because their strategy based on fully understanding of the protein dynamic conformation. And I know about this sentence because I did do some proteins earlier in my PhD work, where I was dealing with a compound called casinolytic protease. It was highly dynamic structure, and we will not be able to detect the structure over 2 years of work. And I know here is the same case in compound in protein SHP2. So the strategy of Relay is to fully understand the dynamic conformation of the protein by fully synthesizing the protein in vitro and using advanced visualization techniques like cryo-electron microscopy and X-ray to identify the dynamic regions in the protein. Based on this, they generated a visual simulation of full proteins, and this will give them the results as we. This is application of the generalized model. They will give them an idea about how to modulate the protein behavior and how to identify a novel allosteric sites that is not occupied by any other ligand. So they used three advanced techniques to do that. The first one is a swimming simulation, which needs a huge cluster. This simulation based on allowing the compounds to move all over the protein to make sure we identify any potential binding sites. But as I said, it needs around 1 million scale per minute, which need a very huge cluster to do it. Next, they used their Dynamo MD simulation approach, which allows them to move from the classical 0.3 microsecond to 10 microseconds. And as you can see here in the classical model, we cannot distinguish between the compound right and left. But using 10 microsecond model, we can distinguish that compound in the left is more stable in the pocket, but compound in the right leave early. Also, they use the iterative cycle at scales. They by this method they can scan around 1,000 compounds at once, so they can easily distinguish between the active, moderately active, and inactive compound. And by this advanced technique, they were able to bring the compound to the clinical trial in around 3 years, and this is some of the promising in vitro data they published in the American Association Cancer Research in last April. So here, as you can see, they have a very novel or a very promising pharmacokinetic profiling of the compound. The compound were able to inhibit the wild type and the mutant form of SHP2, and also were able to inhibit the KRAS wild type and the mutant in a cell model they already invented. Also, they were able to inhibit the MAPK pathway, which is one of the fundamental pathways in the cancer. And in vivo, they showed a very good activity in a dose-response manner in xenograft models. And as I said, the compound now in the clinical trial, actually it finished the phase 1, and right now it will enter the phase 2. So I have just 2 minutes. I will just go over our project in RugMed. It's kind of confidential information, but since the conference theme is about cancer, so I will just disclose some information we have. So one of the fundamental projects in RugMed is to overcome the venetoclax resistance and post-surveillance post-marketing surveillance. So as you know, venetoclax is marketed under the name of Venclexta from Genentech AbbVie. So this compound is used to target the BCL2 protein and is the first PH3 mimetic compound to be marketed. Unfortunately, this compound subjected to resistance in the post-market surveillance in 50% of the cases due to two reasons that we already explored. The reason one is the target mutation. So here is a binding site in the BCL2, in the B1, the thing that was named by the inventor, B1 part, has kind of mutation. So we have 4 different proteins right now. So this mutation weakens the binding of this compound to the protein. And the second thing is that our body is smart, so the body finds a way to drive the survival pathway using another protein called MCL1, which is already present in the mitochondrial surface. So in the mitochondrial surface. So our strategy in RugMed to overcome this resistance is to design a new molecule using ultrafine scaffold hopping to make another molecule that has the ability to bind to the mutant forms of BCL2 protein. Also, we will use the PROTAC approach that Dr. Hilal illustrated in his lecture to overcome or to degrade the MCL1 protein. So our idea is to inject both together to make sure that we don't have any alternative survival pathways. And we also have a molecule that can bind potently to the mutant forms. So this data we collaborate with In Silico Medicine in their headquarters in the Emirates. So we were able to get 10 compounds. So actually we use the ordinary scaffold hopping techniques. We get around 1,000 compounds that has the potential ability to bind to the mutant forms of BCL2. But using the In Silico Band-Omics, we were able to identify just 10 compounds that have a very potent characters to bind here. And also we know that these compounds are patentable, so we are not fear about any intellectual property in the future. And also we were able to identify 5 protein sequences that we will use as CCBL recruiting arm in our PROTAC design. We will not be able to use the SV ligase, the ordinary one, because only CCBL is take advantage of the presence of this ubiquitin ligase, we were able to identify the five peptide sequences. We are working right now on the sense of these compounds and these sequences, and hopefully next year I will have some promising data for you. So to here I will stop, and I will thank you so much for your active reading. Thank you.
Thank you.